now?
Hey! I'm reading up on some racial disparities that are associated with breast cancer, and I'm looking at it at an angle from equity issues as well as the contributing genetic and molecular components. And I'm just trying to tighten it up so I can start putting together my thesis proposal.
Oh, cool. How are you going to test that out?
So I think that if I use a knockdown system to target genes, I can see how that relates to cancer growth. And so to do this, I need to use human breast cancer cells. And then I think I'll use a lentiviral platform to actually make the knockdowns. And what's really cool is if the initial experiments work out, I can move into a rodent model.
That sounds like a really good idea.
Thanks.
But why do you look so worried?
Because it's a lot going on. I have the cell lines and the viral vector and, of course, the mouse system. I'm going to need an IBC protocol, and I've never written one before.
You know what you should do? You should call the Biosafety office and see if they can help you out.
I hadn't thought about that, but that's a good idea.
Yeah, let me know how it goes, all right?
I will. Bye.
Human cell lines and viral vectors, which are considered to be engineered viruses, are biological hazards and require appropriate engineering and administrative controls, as well as appropriate personal protective equipment. All this needs to be planned for when designing your experiment, and a proper risk assessment needs to be conducted. In addition, the institution will more than likely require all biohazardous research protocols to be sent to the Institutional Biosafety Committee, or IBC, for review and approval. This is especially true if recombinant or synthetic nucleic acids are going to be used. It is always a good idea to reach out to the University Biological Safety Officer or the Biosafety Office for assistance.
Hey, Dora.
Hey,
I just got off the phone with the Biosafety office.
how did it go?
They were so helpful. I now know what I need to do to get things moving.
So, what's the plan?
So first I need to talk to our PI and have him help me get an IBC registration form so I can submit that for review and approval. And then she also said because I do biosafety level 2 work, that I need to go online on the CDC or NIH website and find a form called a BMBL.
BMBL?
Yeah, it's short for Biosafety in Microbiological and Biomedical Laboratories. Apparently, it's this form that kind of walks you through the different levels of containment practices for work with biological hazards.
So, what do you have to do for Biosafety Level 2?
I don't really know. I'm going to take the module today and find out.
All right, let me know how that goes.
Sounds good. I'll see you.
Bye.
The first step before initiating a project that will use recombinant or synthetic nucleic acid technology and or biological hazards is to get Institutional Biosafety Committee or IBC approval. The IBC is a committee that is designated to review and ensure safety with any research that uses recombinant or synthetic nucleic acids and other biological hazards. Since viral vectors use recombinant DNA technology and are potentially biohazardous in and of themselves, they require a protocol to be submitted to the IBC for review and approval. Additionally, training is required on BSL-2 practices, and most institutions have BSL-2 training.
Hey!
Hey,
How's it going?
good, just wrapping up.
Nice.
I heard you were starting your project today.
I am. I finally got the IBC protocol approval, and so today I get to grow my cell lines to start to grow my lentiviral vectors. And I watched the lab manager do some culture work yesterday, and so today she's going to watch me do some of mine.
Very cool.
Yeah, I'm excited.
Good luck.
Thanks.
Oh, hey, Haley.
Hey.
Are you ready for experiment today?
You should always wear the appropriate PPE upon entering the BSL-2 facility, and the signage will help remind everyone. Also, make sure that the door is closed and locked behind you.
I am.
Do you mind walking me through it?
Not at all. So I have my procedures and my SOPs in my notebook. My first step today is I need to make my lentiviral vectors. So I'm going to co-transfect my plasmids into my HEC293 T cells. Okay, so I'll go ahead and open the hood.
Sounds good.
All right. And then how long should it run before I do work?
I would let it run about 10 minutes, but
Okay.
you can set up in the meantime.
Okay.
The biosafety cabinet should be run for 10 minutes prior to use. This ensures that any stagnant air has been filtered through the HEPA filters, otherwise known as high efficiency particulate air filters. Additionally, Wiping down surfaces with 70% ethanol while the cabinet is running ensures the air and surfaces are as sterile as possible.
Okay, how does my setup look?
It looks good, but I would recommend putting down some bench pads in case you have a minor spill.
Okay. I think I'm ready.
Looks good. I'm just going to walk you through a few tips as you begin your work.
Okay.
The first tip I can give is remember to not make fast movements while you're in the hood.
Okay.
Also, work clean to dirty. It would be best if you could avoid moving in and out of the hood as much as possible. Additionally, I would say the most important tip is to not leave anything on the grill, because this allows room air to enter the hood and bypass the HEPA filters.
In order for the biosafety cabinets to work efficiently, the user must follow basic aseptic work practices in order to ensure that the experiments are free from contamination and that the user is protected from exposure to any potential biological hazards. That's the whole idea, to protect both the user from the experiment and the experiment from the user. Working in an organized manner will help keep your cells from being cross-contaminated. An important thing to note here is that all waste receptacles are placed inside the biosafety cabinet. This is to avoid moving in and out of the cabinet during the project. In doing so, it can disrupt the protective air curtain, which could potentially lead to both contamination or exposure.
All right, I think I'm done.
It looked all good. Can you show me your cleanup technique?
Yeah.
Proper decontamination of materials and the biosafety cabinet itself is important when working with biological hazards. Each item must be sprayed down with 70% alcohol solution and wiped down upon removal from the biosafety cabinet. All waste containers must also be sprayed down and disposed of properly once finished. The small biohazard bag must be tied securely, sprayed down with alcohol, and disposed of in the appropriate biohazard bin. The sharps container must never be filled past the fill line, so always check it prior to starting work. Make sure that a full sharps container is also wiped down and disposed of through the biohazard waste stream. Finally, it's important to make sure that the biosafety cabinet is wiped down completely before turning it off for the day.